So thank you, happy to be there, and it's really natural that we speak after the previous talk and the questions that were asked. So to start, so our common work with Thibault and myself from Bordeaux, where I am now, and Thibault was working till recently in Vienna. So the question was, is the plant a genetic mosaic? An insight into plants' individuality. So we have already started to talk about this with all of you. And so the title, the second title, could be, can genetic studies change our views about the individual plants? So it's another way to talk about it. Okay, let's stay this way. So first, definitions that we don't really need because we talked about this already. So I have to say again that the definitions are really regularly animal driven, and that's a problem for plants. And so we have to repeat that individual comes from Latin individuum, which means something indivisible. So you cannot cut an individual. So, but we are interested today from the genetics perspective. And so the biological individual can be defined with this perspective by a genetic unity. So the same genome through time is observed for an individual. And by second definition, the homogeneity of its DNA sequences through the space. So the same genome can be observed through the organism space, whatever you look at. So the idea is you don't have much mutation in this picture. So the individual is, by definition, an homogeneous genetic unit. And so you see here the tree with several parts that can be compared. And the question is, are they the same or not? So the plant was historically considered as a population of fragments. It was already said in the previous talk. And you see here all these references from Greek history, so Theophrastus till today, almost, with many different famous or less famous people writing on this topic. And we can say that the bud, we can say, talking from these people, that the bud as a seed is the individual plant. And so the plant is not an individual, but a collection of seed or of bud. So, but that's a view that was really common, and that is still common in the Western way of considering plants, and we will see what's the results of this. So, and all these people that were just listed in the previous slides said that, so the plant is considered as a population of fragments, but very often they compared plants to colonial animals that you can see on the slides with a very nice and well-known pictures from Ernst Haeckel. This marine colonial organism composed of medusa and polyps. And so plants were very often compared to colonial organism as being a colony of buds. And so the question, I just say it quickly, I won't go through further. The questions answered here are the difference between a modular and a unitary organism, the relations between part and whole, and the idea that you can start with cell, then go to the organism, then reach a colony, and you have a continuum between these different levels. So that's just a small point. So if you consider a plant as a genetic community, which is something common, why would plant accumulate more mutations? That would explain why you have a genetic community and not a genetic unity. So the first idea is the absence of a protected germline in plants, which is the center of Weismann's theory. So August Weismann created his germplasm theory in at the end of the 19th century, and it is a way to distinguish animal from plants. So to explain it a little bit more, so his theory distinguished for the animals a soma, which is a the body, in fact, and the germline, which is protected. And you see here on this slide that the green mutations here, green spots, or the blue mutation cannot be inherited because they occur in the body and they are not transmitted to the next generation. Whereas the orange mutation here is transmitted because it is in the germline. But it doesn't occur often because germline is an immortal cell lineage, according to Weissman, that is really protected from mutation. And on the opposite, somatic cell lineage are a mortal vessel where mutations can occur because they are not inherited. So in this animal model, there is no inheritance of somatic mutation. And on the opposite, if you apply this to plants, the plant model, you see here if a mutation occurs somewhere in the plant, it can be inherited and transmitted to the next generation. So the seed would be at the tip of the branches, or if this mutation in green would be inherited as well. So in the plant model, since there are many divisions between the seed giving rise to the plant and the next generation, we can expect that this late germline differentiations give rise to the inheritance of somatic mutations. So that is the big difference between the plant model and the animal model, and that's something we question in our study. So why would plants accumulate more mutation? We saw the first important reason, and there are also the long lifespan, for instance, for trees, open system of growth, multicellular propagation, which would be a reason to explain that mutation would be more common in plants than in animals. And so the mosaicism is expected after this observation, and it's supposed to be more common in modular plants than in unitary organisms, for instance, us, mammals, in older plants than younger ones. So that was the work of Ellen Clark. So in this case, the genetic homogeneity thesis for individuality failed, and that's the problem we are coping with here. So we just wanted to come back to the case study Pando. You see this picture, it was almost the same as the one of Özlem yesterday. And so Pando was cited both by Özlem and by John yesterday. So it's important to come back to this example, which is a paradigm for plant individuality. So it is called the giant aspen clone. It's a quaking aspen, or one of the biggest and oldest plant individual on the planet. And the individual in this case is several trees. So there are almost 50,000 stems on 50 hectares, and only one single genotype is assumed because of asexual reproduction, as you see here, no seeds, but just sprouting that have produced this forest. So for Pando, for instance, how does genetics revise this myth, and how can it help to see it in a maybe different way? So Canadian colleagues did some work some years ago with microsatellite loci, and so they genotyped all these trees you see here, a bird eye view of Pando. And what they observed is that indeed there is very common genotypes, which as they call it Pando genotype. In green here, you see it. There are many trees here corresponding to this genotype, but there are many other genotypes present around and even inside in some places. You see the other colors. So there is not a unique genetic individuals in concerning Pando. So you have to revise the way you consider it, even if it's true that the clone is big and that the same genotype is present. There are many other colors, and these colors are due to sexual reproduction, probably. And other observation among this Pando genotype, the green one, they found very rare somatic mutations, so many small mutation inside this genotype. So that suggests that the clone is not so old as expected, and that it's more recent because there are not many mutations. So I just finished this example with the citation of Ellen Clark in 2011. There are known aspen genetics that are 50,000 ramet strong and estimated to be over 10,000 years old. So this sentence has to be revised according to genetic results. So where do we find somatic mutation? That would be one question. I can show you here some agronomic importance of somatic mutations, because we find somatic mutation in cultivated plants, and they are special plants. So the somatic mutations are still rare, but they are facilitated by vegetative propagation, and they are selected for by humans when they are interesting. So for instance, here, a common example of grape vine, you know, Pinot Noir, Pinot Gris, Pinot Blanc, you may know this species, and they are deduced from one to the other thanks to transposable elements which are really favored by vegetative propagation. You know that it's the way we cultivate, we cultivate grapes. So here you see the effect of somatic mutation in a very specific way. And there are many, second example and last one, many new fruit varieties are issued from bud mutations, or also called bud sports. So they were not obtained through seeds, but on a tree, for instance here, a peach tree on which suddenly a branch gives nectarine. And it was observed several times at these times. You see here the references. Or a grapefruit in Florida was observed apparently only once in 1906. Pink grapefruit occurred on a yellow grapefruit, a classical one. So that's something that happened, and that's true somatic mutation. So just to conclude with the wild phenotypic mosaic, we just have one example to give to you of a wild plant with a phenotypic mosaic. You see here the yellow circles. They correspond to herbivore-resistant branch that was observed in Australia on a single tree, which is quite famous. But we don't have much to say about this tree. It's quite a strange story. But we don't have other examples to give of wild plants showing mosaic. So that's it. So our question was to check the reality and importance of intra-individual variability in plants, and we have some examples to give to you that we found in literature, in published papers. So the tree geneticists, that's Thibaut and myself are both tree geneticists. What we do is to sample material anywhere in a tree, represented here. Leaves, phloem, cambium, buds, that can be any part of the tree that you can extract DNA from, and then you extract DNA and you sequence it. And the simplest thing you can look at is the simple genetic variation is SNP or SNPs, single nucleotide polymorphism, here represented by the 3 branches and 3 sequences. And you see in red that nucleotide is modified compared to the other branches. That's the kind of markers you use when you sequenced, for instance, different parts of the same tree. And here, that's the extraction of DNA some times ago. So what kind of studies were done with this kind of analysis? So maybe you heard that Napoleon was died yesterday 200 years ago. So this is an example of a tree still alive, which is called Napoleon, like the guy, maybe less dangerous. So you can find this tree on the Lausanne University campus. They sampled 2 parts of the trees you see on 2 different on top and on base of the tree, and what they found comparing the whole genome was only 17 somatic mutations, so 17 SNPs, as I told you before. So since the oak genome is quite big, 750 million base pair, the percentage of somatic mutation found between these 2 places, extreme places of the tree, which is 250 years old, is very, very low. So this percentage has not high meaning. It's just to say that differences that were found are really very low. So second study, which was performed in my lab, in the lab where I'm working and where Thibault was a postdoc before, about another oak that you see here, which is 100 years old, a little bit younger than the previous one. 3 branches were studied and 46 somatic mutations in the whole genome were found. And the second interesting thing was some acorns were collected and somatic mutation seems to be transmitted to the next generation. They observed some somatic mutation still present in the acorn. You see here a picture of the tree with where they do crosses, it's controlled crosses on the tree, which is something difficult to do with trees, but it's done. So other example that we found in a crazy paper from Wang et al. So in 2019, evaluating within plant variation with lots of sample. If you want to discover a very difficult paper to read and so rich paper, you have to read it. So, and we give some example of the paper, there are many. So here, patterns of somatic mutations in leaves and in the roots of a peach tree that is represented here. So samples were taken from the tree. You see the blue tips here representing samples, and also in the roots, like here. And the new mutation observed from samples to sample are represented in yellow here. And they did some phylogenetic trees to study the distances between the leaves and between the roots. And you see that the tree is really longer for roots than for leaves, which suggests that there are many more mutations in roots than in leaves. So these two tissues are different concerning mutation. So that's something you can say after the study of these Chinese colleagues. Another study they did, another species they studied intensively was the patterns of somatic mutation in woodland strawberry runners. So they took some leaf samples and some runners samples, as you can see here. And what they found, I don't know if you can see, but you see here, the yellow 1, 3 means that the mutation numbered 1, 2, 3 were found here for the first time. And they were always found on the runner, but never on the leaves, on the shoots coming from the runner. So that was an interesting discovery. So there are mutations specific to the runner and mutations specific to the leaves. So that's quite surprising for a plant. So conclusion, first slide of conclusion over 2. There are differences between plant species. For instance, the heritable mutation that were observed for oaks, as I told you before, maybe there are more heritable mutation in perennial than in annual plants. So that's first result that has to be confirmed. There are differences between plant parts. Some plants tissue accumulate more mutations, for instance, the roots and the shoots of peach, as you saw before. And there seems to be specialized cell lineages, we can call them this way. So runner-specific mutation in strawberry, just what that I showed you before, and germline with slow division in Arabidopsis and tomato, which are illustrated by this figure where the number of division is low for blue parts, and you see that there are blue parts in the center of the bud, which seem to be protected, so less mutation occurring in them. So Weissman theory, as I presented to you earlier, would be valid for some plant species. So that's really something interesting and that nobody could guess before. So that's the first result, interesting result. Second slide for conclusion. Our initial question, is a plant a genetic mosaic? There are degrees in mosaicism, we can find them, but there are only few point mutations and some are inherited. That's what we saw. But globally, there are no accumulation of somatic mutations that were observed in the different studies. And so plant trees are not highly heterogeneous. And so by consequence, the plants and not the bud fit the individual genetic definitions in opposition with the history I showed you at the beginning. But what we have to say here is that only the simplest mutations, SNPs, or SNPs were studied in the different articles we showed you, and plant genome are more complex than only simplest mutation. The structure is complex, its composition, and you see here a representation just quickly shown of rice genome. There are, of course, genes, there are transposable elements, there are a lot of transposable elements, there are retrotransposons, so there are many other kind of things composing the genome, and we don't have always information about intra-individual modification of this kind of components. So, pour finir, so to end, last slide, it seems that genetic, or at least we think that genetic homogeneity, heterogeneity are good tools for individual studies, but there are not so many publications available on this question. And it can help to revise some received wisdom in philosophy of biology, for instance, Pando that I showed you. But plants, as other non-vertebrate species and colonial animals as well, refuse too broad generalization in French, and call for a plurality of individuality approaches. So individuality is a matter of degree. That's not a new idea. It was developed by several philosophers. And we started to talk about this subject just in the previous talk. So our idea with this presentation was to bring plant science and philosophy closer and to cut the mustard, which is a nice British expression, on plant world puzzles. Thank you.